My name is Nick Turner. I work for the Spatial Data Analysis and Visualization Lab. We are under a EPSCoR grant, and right now I've been working with Dr. Price and Dr. Stacy on creating some images from the Scanning Electron Microscope. One of the projects I'm working on is the Metrosideros polymorpha. It's a ohia leaf, and we are looking at the trichomes, the hairs of the ohia leaf, and comparing the different characteristics of different species with each other under the SEM.
I'm Dr. Elizabeth Stacy, and I'm Professor of Biology and TCBS here at UH Hilo. And I'm an evolutionary biologist, and I'm fascinated by where plant species come from, especially trees, because there are at least 60,000 species of trees in the world, and we know very little about how they got here. So Hawaii is a great place to study speciation, and especially with Metrosideros, the most common tree here in Hawaii, using that as a group to try to understand what drives speciation and how long it takes and those sorts of things.
The other project I'm working on is the Drosophila picture wing flies, and the flies have to be prepared in a different way than the ohia leaves. The flies are a lot more delicate because it's an actual living insect, and so what we do with that one is we put it into a critical point dryer, which is the machine you see right there. And with the critical point dryer, we actually are able to dehydrate the fly without actually losing any of its physical characteristics.
I'm Dr. Donald Price, and I'm a professor of biology and chair of our Conservation Biology Environmental Science graduate program. And what we're doing here is I study some insect species in Hawaii. They're a very fascinating group, Hawaiian Drosophilids. It's about a third of the world's Drosophila species live in Hawaii, so we're all fascinated about why a lot of these species come about. And there's a group of them that are the picture-wing Drosophila. These are Drosophila that are quite large for Drosophila. They're about the size of a housefly. And as their name says, they've got picture wings. So they've got usually a patterned wing that's fairly species specific. So you can actually tell the species out in the wild fairly well. And then they have a lot of other differences between species. So some of them have large morphological differences, behavioral differences, and we look at their behavior and their genetics of how do they stay as different species, how have they speciated in the past, how do they adapt to their natural environment here.
One of the amazing things that the SEM allows us to do is get down to a cellular level of magnification that you just can't do with any kind of a conventional microscope. And so this allows you to surpass the limitations of a normal digital camera or even a microscope itself.
Nick is doing some fabulous work with Metrosideros, and what his work is allowing us to do is to see these different forms of Metrosideros, obviously close up and personal. So they tend to differ from each other in leaf characters, so the shapes and sizes and pigmentation of the leaf, but also the pubescence. And so it's really hard to see some of these differences with the naked eye. And a lot of these forms don't even have official names, official taxonomic names, but we can keep seeing them repeatedly in nature. And so what Nick's work is allowing us to do is to quantify on a very microscopic scale some real differences amongst these things. So it's kind of neat.
So two that Nick is helping us with the electron microscope are two species that occur here on the Big Island of Hawaii. And they're very different morphologically, but they can hybridize, so they can produce hybrid offspring. So we're looking at the genetics of that. We're looking at the behavior of these different species. How are they staying separate? We know they must be genetically different because they're morphologically and behaviorally different, and that has to have a genetic basis, but we really don't know why they can hybridize, even though they're distinct species. And they can actually produce what we call advanced backcross and advanced hybrid offspring. So we're looking at sort of these multiple generations hybrids and what's causing them to stay separate but still able to hybridize to some degree. That's one project. Then the other project that we're working on is another species that's pretty widespread around this island and has large elevation gradient changes. in it. So we're looking at the adaptation of these species to the climate change that's occurring, temperature changes that particularly occur in this really common species, and then comparing it to some of the more rare species, because some of these Hawaiian picturing Drosophilas are endangered species, believe it or not, for an insect. And we don't really know why. We know that some of their host plants have declined. There's been some invasive species that have come in, and temperature is another possibility for why some of the species are changing in their abundance across the environment.